Hello everyone, glad to see you. My name is Alexandra Shakira Hadi, and I would like to present my research proposal with title Screening of Oxazole Derivative as Antitubercular Candidate Through Inhibition of Polycated Synthase 13 Enzyme Activity using in silico method. And here's my outline presentation. So now we start from introduction. And this picture is when Englanders blamed vampire for tuberculosis death, and it happened before the discovery of tuberculosis. Tuberculosis is one of the oldest human diseases, having co-evolved with humans for hundreds or maybe millions of years, until in 1882 Koch revealed the discovery of TB pathogen at the Berlin Institute for Physiology. So that we know, the bacteria causes tuberculosis is Mycobacterium tuberculosis, which commonly affect the lungs but can affect practically all organ systems. Despite being an ancient disease, millions of people worldwide still suffer and die from TB disease. Compared to HIV or any other infectious disease, TB has substantially greater annual mortality rates. According to WHO data in 2020, 10 million people are infected with tuberculosis, and in addition, 1.5 million people died from TB, including 214,000 people with HIV. India, China, Indonesia, Philippines, Pakistan, Nigeria, Bangladesh, and South Africa are the countries that contribute to most new TB cases in 2020. Drug resistance is one of the causes that make TB disease is hard to eliminate. Therefore, research on the discovery of new drug candidates needs to be carried out. So, in this research, oxazole derivative will be screened as inhibitor of the PKase13 enzyme, and I hope that there will be oxazole derivative compound that can be used as candidate for antitubercular drug. PKase13 plays important roles in mycolic acid synthesis, and mycolic acid is component of the cell wall that have vital roles for MTB life defense and pathogenicity. Anaxazole is potential compound that can be used as a drug candidate seen from its unique structure. So here's a problem identification of this research. So the first one is there any oxazole derivative compound that potential to be inhibitor for PKase13 enzyme? And the second is how is the interaction between inhibitor ligand or oxazole derivative and the PKase13 enzyme? And the last is do the result of ADMETOX study on selected oxazole derivative compound as a drug candidate have a good value. So now we move to the aims of this research. So the first is obtaining novel PKase13 enzyme inhibitor that are effective and can be utilized as a drug candidate based on oxazole derivative compound. And the second is find out and analyze the interaction between oxazole derivative compound and PKase13 enzyme. And the last is performing data analysis for molecular docking simulation result and ADMETOX studies as a parameter. So now we move to hypothesis. So here is the hypothesis I propose. The first is find out and analyze the interaction between oxazole derivative compound and the PKase13 enzyme. And the second is the PKase13 enzyme interacts well with the selected oxazole derivative compound. And last is the ADMETOX study on oxazole derivative compound found it safe to utilize as a drug candidate. So to know and understand more about this research, we move to literature review. Okay, so the idea of this research are coming from several previous studies. So here's state of the art. Um, In 2017, Agrawal et al. Have confirmed that TAM16 is a potential compound that can be used as inhibitor for PKase13. And in 2019, Sinaroglu Antimusin has done virtual screening and have confirmed that compound with benzofuran ring is a potential to be inhibitor PKase13. And in the same year, Arba et al. Has done virtual screening to 107 small compounds and found that there is a four potential compound can be used as PKase13 inhibitor. And in 2021, PDEP et al. Identified 20 enzymes that have an important role for Mycobacterium tuberculosis growth and defense as a target for substitute oxadiazole compound and found that PKase13 is the most potential target. So for tuberculosis disease is an infectious disease caused by MTB, which generally infect the lungs but can affect almost all organ system. From a clinical and public health perspective, people with TB are pragmatically classified as having latent TB infection or LTBI, active TB disease, and subclinical TB disease. So now we move to Mycobacterium tuberculosis. As we know, MTB is an aerobic, non-forming spore, non-motile, or don't have ability to move bacillus with a cell wall containing high molecular weight lipids. And MTB transmits from one person to another through the air, and humans are the only host where MTB can live and reproduce naturally. MTB is pathogenic bacteria that grow slowly. MTB takes 15 until 20 hours to grow, whereas other harmful bacteria take less than an hour. And this picture is a map of complete genome sequence of the H37RV strain. A comprehensive understanding of the mycobacterial genome began in 1998 when the complete genome sequence of the H37RV strain was discovered and analyzed. And this genomic information play an essential role in translating gene expression, modeling persistence, and identifying essential protein that can be used as a target for drug candidate. So for polycated synthase 13 or PKase13 is an essential enzyme in mycolic acid synthesis in MTB, and this enzyme works to catalyze the condensation of two fatty acid chains to form a ketoester as a final intermediate of mycolic acid, and it consists of five domains. The medial three are the mandatory polycated synthase domain. There are the ketoacyl synthase or KS domain, the acetyltransferase or AT domain, and the acyl carrier protein or SCP domain. The other SCP domain is adjacent to KS domain and the thioesterase or TE domain at the C terminus of PKase13. And this picture is reaction mechanism, how PKase13 works. So now we move to mycolic acid. Mycolic acid is a long chain fatty acid found in cell walls, lipid-rich mycobacteria such as Mycobacterium tuberculosis. And mycolic acid is composed by beta-hydroxy fatty acid with a long alpha alkyl side chain. An enzyme involved in its production are promising therapeutic target, and mycolic acid plays such a vital role in survival and pathogenicity. And this picture is MTB cell walls, and this is the five structure of mycolic acid. So now we move to oxazole. Oxazole or 1.3 oxazole is five-member heterocyclic ring structure containing an oxygen in the one position and a nitrogen and in the three position and both separated by carbon. And several studies have stated substitution pattern of oxazole derivative has biological activities that have important roles such as antimicrobial, anti-cancer, anti-tubercular, anti-inflammatory, anti-diabetic, anti-obesity, and antioxidant. So now we move to in silico method. The term in silico is a term that refers to computer-assisted experimentation. And computational or in silico methods are knowledge-driven, explore rationally available data to investigate protein function and design new molecular entities that can effectively regulate protein behavior. So this figure is an example of an in silico drug discovery pathway. So now we move to material and methods. This is the material I use for this research. The first one is the PKS13, and the PKS13 protein 3D structure was retrieved from the RSGB protein data bank and downloaded as PDB format. And then I also use oxazole derivative compound and obtained from the National Center Biotechnology Information or NCBI, and compound were downloaded and saved as SDF format. This is the software I use for this research. So the next is research methodology. The first thing I do is I retrieve protein target from protein data bank in PDB format. And the next is I do protein preparation using MOE, and I retrieve the ligand from PubChem. And the next step is screening ligand based on its toxicity and Lipinski rule, and using O-series data warrior. And then the next is screened ligand prepare using MOE. And the next step is an important procedure of this research. It is a molecular docking simulation to see and analyze the interaction between PKS13 enzyme and oxazole derivative as ligand. And the parameter used during the simulation process are the value of delta G binding ligand and RMSD. And the selected ligand is the one with the lowest delta G binding value and RMSD below 2 Armstrong. And then the next step is the most important step, because this research is related to the discovery of new drug candidate. Uh, the most important step is the ATMETOC study, and this research I use and in this research I use ATMET-SAR and Swiss ATME to test the drug likeness and pharmacokinetics of ligand, and I also use TOX3 to investigate ligand mutagenicity and carcinogenesis. So now we move to, okay, so this is the timeline of this research, and here's references I use for this research. And thank you for your attention.